In 1870, Edward Hitzig and Gustav Frisch performed experiments on dogs by which they were able to produce movement through electrical stimulation of certain parts of the cerebral cortex. This was a milestone in the controversially discussed issue of cerebral localization. Before Hitzig and Frisch, there was a man named John Jackson. He was a British neurologist in the year 1861. He was interested in the cortex as an important center for the initiation of movement. Hitzig and Frisch provided the experimental support for Jackson's hypothesized motor area in the cortex. How did the experiment go? It was a bit graphic, but normal for the time. The two restrained live dogs and, without anesthesia, cut away the dogs' skulls to expose an area of cortex. Then they stimulated that cortex with a current from a battery. This caused movement of the dog's contralateral muscles and produced movement in a predictable way. The two finally had experimental evidence for the motor cortex. In 1917, Charles Scott Sherrington, an English neurophysiologist, experimented on chimpanzees, orangutans, and gorillas. He tested different parts of the motor cortex with electrical stimulation to better understand the effects of electrical stimulation and make a functional map of the brain with electrodes. He looked into everything from eyeball movements to face and tongue movements, and to larger areas like arms and legs. He mapped which part of the cortex was related to which movement. In 1920, Walter Rudolf Hess, a Swiss neurophysiologist, implanted electrodes under general anesthesia. Chronic electrode implants were developed by Hess. Chronic subcortical electrode implants were implanted in lab rats to deliver electrical stimulation to the brain. These were also called focal brain stimulation, which is a form of electrotherapy. In 1930, Wilder Penfield redrew the map of the brain by opening the heads of live patients. He was a Canadian neurosurgeon and was known for treating epilepsy. He created a procedure called the Montreal procedure. This helped him to pinpoint the source of the seizures in the brain so that he could remove that area of the brain during his surgeries. The patients stay calm and awake during this procedure. Wilder Penfield would probe the surface with a small electrode, and by probing certain areas, he could have results such as numb fingers, suddenly retrieved memories, flashes of light, or smelling a certain odor. If Penfield found the area that produced an aura which is generally associated with epilepsy, he removed it, and this reduced the risk of seizures.